Sci Talk is an online webinar series conducted by the Botanical Society of University of Colombo. Botanical Society of University of Colombo, also known as BOTSA, is a student body that's established under the Department of Plant Sciences, and it's one of the oldest and most active student bodies in the Faculty of Science. BOTSA has been able to conduct various projects like community-related projects, workshops, and webinars with the aim of increasing the awareness and interest for plant sciences while providing a platform for its members to improve their soft skills, just like what I'm doing now. So, the Sci Talks webinar series was initiated during COVID pandemic as an online platform to share the knowledge and experience of our very own alumni members. Through Sci Talks, our current undergraduates get an opportunity to grasp a lot of knowledge and insight on the possibilities of what they can achieve in future. So today, as the speaker of 9th session of webinar series, I warmly welcome Dr. Ifthiha Mohideen, a postdoctoral fellow at the University of Alberta. She has completed her PhD in biology at Concordia University, Montreal, Canada in 2021. During her PhD, she worked in the lab of Dr. David Kwan, where she focused on researching and harnessing the substrate flexibility of enzymes involved in natural product glycosylation towards better therapeutics. Before joining Concordia, she obtained her BSc with a first-class honors in plant biotechnology from the University of Colombo in 2014. She was a recipient of the Professor B.L.T. De Silva Memorial Prize in Plant Sciences, Dr. Swarna Sentiraja Memorial Prize, Sir Nigel Ball Award for Plant Sciences, and of course, the Department of Plant Sciences Staff Prize. After the graduation, she worked as a demonstrator and an assistant lecturer at the Department of Plant Sciences and Research and Development Specialist at Colombo Science and Technology Cell. So, without further delay, I would invite Dr. Ifthiha Mohideen to conduct her presentation for our enthusiastic audience on an engineered enzymatic pathway towards anticancer anthracycline glycosylation. Here's a kind request. To minimize any disturbances during the session from the audience, please switch off your videos and mute yourselves. If you have any question, please wait until the Q&A session at the end of the presentation. So, Madam.
Thank you, Sarala, for your kind introduction. Let me share my screen. Okay, can you confirm whether you're seeing my screen?
Yes, Madam, we can see.
Okay,
but
perfect.
Still the presentation mode.
Yeah, I know.
Oh. Okay.
Okay, perfect. Yeah, good evening, everyone, and it's so good to be back with the place that I started, even virtually. So first of all, I would like to thank Dr. Chandrika Nanakar and also BOTSOC for giving me this opportunity to share my research work with you all. And before I move into the actual science, I thought of walking you all through my scientific journey. Maybe it will be an inspiration for some of you. So I did my bachelor's in plant biotechnology at University of Colombo, Sri Lanka. And then for higher studies, I moved from Sri Lanka to Canada. And to be specific, I moved to Montreal. And in this map, you can see this is Montreal because I started a master's in biology at Concordia University. And then after 1 year, I fast-tracked into the PhD in biology. And then last summer, I was able to complete my PhD program after many years of hard work. And then I moved from Montreal to Edmonton. So if I just tell you the distance between these two cities, it is more than 3,500 kilometers. So I took the move because I got a postdoctoral fellowship at the Department of Chemistry, University of Alberta. Okay, so today I will be presenting a part of my PhD research work, and the title is An Engineered Enzymatic pathway towards anticancer anthracycline glycosylation. So when I tell this title, I know that many of you will not understand this at all, but that's totally fine. By the end of my presentation, I will make sure that you will learn something important. And also, before I move into the next slide, I would like to mention that even though I did my PhD in biology, my research project is not complete. It's not exclusively in biology or biotechnology. It is interdisciplinary, which means you will learn concepts and techniques in the fields of synthetic biology, molecular biology, and even biochemistry. So uh, you all know what is this, right? It's honeycomb, or in Sinhalese we call it miwade. So honeycomb is made up with beeswax, and obviously it contains honey. So the main composition of honey is fructose and glucose, and we call these molecules as sugars. So what is the importance of honey? So you might say that from the ancient ages, people have used it as a preservative, right? So yeah, that's true. There are a number of applications of honey, as well as other sugars and sugar containing molecules. And also when I mention the term sugar, it doesn't mean the regular table sugar or honey only. So the term sugar means a number of other molecules, and you will learn about it in further slides. So in my PhD project, I became interested in small molecule natural products with sugar moieties. So many of these small molecule natural products are produced by a number of different organisms, including bacteria, fungi, algae, and also plants. And these are produced through their secondary metabolic pathways. And interestingly, many of these molecules are decorated with one or more sugar moieties, and these have a great effect on their biological activity. So why are we interested in these sugar-containing molecules? What's the importance? Why we bother? Because many of these sugar-containing molecules or glycosides have therapeutic importance. So here I have shown the well-known antibiotic structures, erythromycin and vancomycin. So you might think that I'm going to give you an organic chemistry lecture. No, this is just an illustration to show the structure of these molecules and where these sugars are attached. So this is erythromycin, and there are two sugar residues in erythromycin, and this is vancomycin, and it also has two sugar residues. So basically these are glycosides. On the other hand, we can also find anti-diabetic agents such as montbredin A. It is also a glycoside, and it has 5 sugar residues, and also the anti-cancer drugs daunorubicin and doxorubicin. These are also glycosides, which means each of these molecules has a sugar residue. So as you all can see, these chemical structures are highly diverse, and so does their function. So how are these molecules produced in nature? So before I answer that question, let us go back to our previous example on honey. So if we focus on the production of honey, we can identify 3 distinct elements. We can identify the starting material, which will be the nectar, and then we can identify the final product, which is honey, and the driving tools, or the organism that is responsible for synthesizing honey will be honeybees. So basically, honeybees will convert nectar into honey, or in other words, they will convert the starting material into the final product. Similarly, when it comes to the glycoside production, there will be one or more starting materials, and there will be one or more products, and there will be a driving tool. And the driving tool of small molecule glycosides are enzymes, So basically, if we focus on the small molecule natural product like biosynthesis, we can identify two classes of enzymes. The first class would be sugar biosynthetic enzymes. As the name implies, they are responsible for the synthesis of sugars, right? So here I have shown, starting from a common sugar precursor, these sugar biosynthetic enzymes will produce a particular sugar donor through a series of enzymatic reaction. So in this enzymatic reaction, this will be the starting material. We call it as a substrate, and this will be the final product. And then the second important class of enzyme include glycosyl transferases or GTs. So again, as the name implies, it will transfer a glycosyl moiety. So for glycosyl transferases, we basically need two substrates. So we need a sugar donor substrate, and also we need a sugar accepting substrate. So basically what this enzyme does is that when it has an activated sugar donor molecule, it can transfer this sugar residue onto the acceptor molecule. Just give me 1 second. Okay. So it will transfer into the acceptor molecule, and then it will produce this final product, that is a glycoside. So an interesting feature of these sugar biosynthetic enzymes as well as glycosyl transferases is that they are substrate flexible. So what does that mean? That means apart from its native substrates, it can act on a variety of different non-native substrates, and it can produce different glycosides. So these enzymes are important in terms of glycodiversification. So glycodiversification, or in simple terms, it means altering the glycosylation, uh, requires these two classes of substrates flexible enzymes. So we all know that these are building blocks, right? But let's pretend these are not building blocks, but these are a combination of sugar donor substrates and sugar accepting substrates. What if we have a substrate flexible glycosyl transferase enzyme? So basically, the enzyme will act on these substrates and it will produce a product, which is a glycoside. But remember, I told you that these enzymes are substrate flexible. Therefore, this glycosyl transferase enzyme can act on some other substrate and it can produce a second glycoside. Or else it can even produce a third type of a glycoside, so and so forth. So as you all can see, these three molecules are structurally different, right? And so does their function. So in this slide, I have illustrated glycodiversification using substrate flexible GTs and sugar biosynthetic enzymes. Just to reiterate, when we have substrate flexible glycosyl transferases, it can act on sugar donor molecules and sugar accepting molecules, which might be non-native, and it will produce altered or novel glycosides. So why are people interested in producing these altered or novel glycosides? Because when you change the structure, the function also gets changed. So in that case, when you alter or when you modify the structure of a glycoside, or when you produce a totally new glycoside, you can also get a totally new biological activity, or you can also get an altered biological activity. In the same way, if you have substrate flexible sugar biosynthetic enzymes, these can produce novel or altered sugar donor molecules, which can also be used by these substrate flexible glycosyl transferases. Okay, so out of the available small molecule natural product glycosides, in my PhD, I became interested in a class of molecules known as anthracyclines. So daunorubicin and doxorubicin are anticancer anthracyclines, and in nature, these are produced by Streptomyces species. So daunorubicin is used to treat acute lymphoblastic leukemia and myeloblastic leukemia. On the other hand, doxorubicin is reported to show broad spectrum anticancer activity. That means it can be used to treat a number of cancer types, including blood cancers and solid cancers such as breast, lung, and ovarian cancer. Despite the clinical importance of these drugs, prolonged administration results in dose-dependent cardiotoxicity, and that's not what we want. If we are using a drug, we want it to have the best activity, or at least to reach the best activity and with minimum side effects, right? So as an alternative to these drugs, researchers came up with another anthracycline analog that is known as epirubicin. So a great feature of epirubicin is that it shows similar antitumor properties as doxorubicin, but it shows reduced cardiotoxicity. So what makes this difference when it comes to the cardiotoxicity? So let's have a closer look at the chemical structures of these two molecules. So this is the structure of doxorubicin and this is the structure of epirubicin. And if I focus on anthracyclines, basically we can divide it into two main parts. So this will be the A-glycan or the non-sugar portion, and this will be the the glycosyl portion. So in doxorubicin, the glycosyl moiety is known as daunosamine, and in epirubicin, it is known as eicosamine. So by now, I believe that you might have an idea about the production of a glycoside, right? So in my previous slides, I mentioned that we need two substrates. We need a sugar donor and we need a sugar accepting substrate, and we need the enzyme. So the sugar donor substrate of doxorubicin is known as TDP daunosamine, and I have shown the structure here. And for epirubicin, it is known as TDP eicosamine. So doxorubicin and daunorubicin is produced in nature, so it is mainly produced by a number of different Streptomyces species. However, epirubicin and its sugar donor is not found in nature, which means a natural biosynthetic pathway for these two molecules is not reported. So conventionally, epirubicin is produced uh, starting from doxorubicin or daunorubicin, and it's a semisynthetic production, whereas TDP eicosamine is produced chemically. Okay, so okay. So the daunorubicin doxorubicin biosynthetic pathway in Streptomyces pyocereus has been extensively studied, and I will just give you a brief overview on it. So for simplicity, today I will be dividing it into three main steps. So the first step would be to produce the sugar donor substrate, that is TDP daunosamine. So starting from glucose 1-phosphate and using 6 sugar biosynthetic enzymes, TDP daunosamine is produced in this bacterial species. So the TDP4 keto 6-deoxy-D-glucose that is shown here is known as a key intermediate that is not only found in TDP daunosamine biosynthesis, but also it is found in a number of different other sugar donor production pathways. And then when it comes to the A-glycan biosynthesis, starting from 1 propionyl-CoA unit and 9 malonyl-CoA extender units, through a series of enzymatic reactions, epsilon rhodamycinone, the A-glycan of the daunorubicin doxorubicin is produced. So it should be noted that fermentation of, Streptomyces fusarius will produce a significant amount of rhodamycin A glycan. However, the production of TDP downosamine in this organism is reported to be very low. So now that bacteria has the A glycan and also it has the sugar donor. So then what it does is that it will glycosylate using a glycosyl transferase enzyme to produce the first glycoside of doxorubicin doxorubicin biosynthetic pathway. And then further enzymatic modifications will produce doxorubicin and its derivative doxorubicin. So being inspired by the doxorubicin doxorubicin biosynthetic pathway, researchers have tried to produce epirubicin and its precursor in vivo. So if you are not familiar with the term in vivo, it means in an organism. So it is a great alternative to the existing chemical synthetic methods. And it has become feasible because of the substrate flexible sugar biosynthetic enzymes and glycosyl transferases. So just to remind you that, as I mentioned you previously, these sugar donor molecules, the main difference is between these molecules is in the C4 hydroxyl group. So TDP downosamine has an axial hydroxyl group, whereas TDP ecosamine has an equatorial hydroxyl group. I just remembered that I forgot to mention it. It's the same for doxorubicin and epirubicin. It's the sole difference is at this hydroxyl group. So this axial and equatorial hydroxyl group difference occurs at the last step of the sugar biosynthetic pathway. So in TDP downosamine biosynthetic pathway, there is this enzyme known as TNMV ketoreductase. So a ketoreductase is an enzyme that can reduce this ketone group and it will produce a hydroxyl group. So this enzyme is known to be stereospecific. That means it will only produce an axial hydroxyl group. That's how TDP downosamine is produced. So what researchers did is, in engineered bacterial systems, they tried to replace this enzyme with some other substrate flexible ketoreductases, which will produce an equatorial hydroxyl group. And in these three papers, they have described it very well, and they were able to produce TDP ecosamine in vivo. But they never isolated it. All they did is they simply used it towards the production of epirubicin that also has this equatorial hydroxyl group. So towards producing epirubicin, the organism now has TDP ecosamine, and also epsilon rhodamycinone is either produced by the organism itself or it can be exogenously provided, and then they used substrate flexible glycosyl transferases to catalyze this reaction, which will produce epirubicin. So even though they were success in producing epirubicin in vivo, so they have constructed a pathway to produce epirubicin in a biological system. However, the yield of epirubicin is reported to be very low. One of the main reasons could be that this enzyme, even though these are substrate flexible, these two substrates are not their native substrates. Therefore, the catalytic activity of this enzyme with these two substrates are reported to be low. So it would be great if the nature had an enzyme that can catalyze this reaction efficiently so that we can produce epirubicin in an organism instead of going to the semi-synthetic process, right? Anyways, in my PhD project, one of the main chapters of my thesis was to produce TDP downosamine, TDP eicosapent and some other valuable sugar donors in vitro using enzymes. So here I have mentioned another term, in vitro. So previously I mentioned in vivo, that means in an organism, and in vitro means outside of a living organism, or in simple terms, in a test tube. So here we don't have living organisms. So why did I become interested in producing these sugar donors and purifying it? Because these sugar donors can be used as precursors or substrates to produce some other valuable sugar donors. This can also be used in glycodiversification, and also it can be used to characterize, co-crystallize, and engineer enzymes. So in 2021, we published this manuscript in SES Chemical Biology, and its title is In Vitro Reconstitution of TDP Downlosamine Biosynthetic Pathway Provides Insight into Anthracycline Glycosylation. So today I will be only highlighting some of the key findings. So if you are interested, you are welcome to read this manuscript to know about the interesting science that we did. So we have reconstituted an enzymatic pathway for the production of TDP downlosamine based on the natural biosynthetic pathway of TDP downlosamine. So basically the reaction I have divided into two main schemes. In scheme A, first we try to produce TDP 4-keto-6-deoxy-D-glucose. Previously, I mentioned that this is a key intermediate, and this can be used towards producing other valuable sugar donors. And then starting from that key intermediate, we call it compound 6. This pathway leads to the production of TDP downlosamine. So taken together, this pathway involves 8 enzymes. So remember, this is done all in vitro. So we are not using an organism. Instead, we are putting everything into a test tube, and then we are performing the reaction. So in that case, first we wanted to get these enzymes in purified form. So how did we do that? So okay, so first we did genomic DNA extraction from the respective organisms, and then we PCR amplified our desired genes that are encoding these sugar biosynthetic enzymes. And then we cloned our gene using a technique called restriction, digestion, and ligation. If I remember correctly, in your third year, you will learn this. But just to make sure that we all are on the same page, I will quickly go through it. So restriction enzymes are a class of enzymes. So these enzymes can go and it can recognize a specific DNA sequence, which is shown here, and then it will cut at a specific site. So we use these enzymes to cut the ends of our PCR amplified genes, and also to cut a vector. In this case, the vector will be a bacterial plasmid. Now we have our cut gene, and also we have our linearized plasmid. Then we use another enzyme known as DNA ligase, and then we paste these ends together to form this recombinant plasmid. And then we wanted to make sure that our plasmid contains our desired gene. So how did we do that? We sent it for sequencing, and we made sure that each of the plasmid contain these desired genes. And then we wanted to express and purify these sugar biosynthetic enzymes, or these proteins. So basically, we introduce these vectors into a bacterial system that is capable of expressing these proteins. So the bacteria does the job, it will produce the protein, And now we have these proteins inside the bacterial cell, right? So we need to get it out and we need to purify. So we lyse the cells and then we use a purification method that will purify your desired protein and remove all the other proteins that might have expressed in this bacterial system. So how do we do that? Basically, the protein of interest, we have tagged it with histidine. So these histag proteins can be bound to a specific resin. So what we did is we lyse the cells, we add the cell lysate to this column, and it has the resin. So all the other proteins that do not have this tag will get washed away, but then the proteins with the tag will go and attach to this resin. And then we elute it using an elution buffer. And finally, we have our protein of interest. And next, we confirm these proteins by running a gel. So this is a protein gel. This is the molecular marker. And in this gel, I have shown all 8 proteins that are used in my reconstituted pathway. So now that we have all our proteins ready, then we started producing compound 6, the key intermediate, using a previously established procedure with necessary modifications. So basically, starting from glucose 1-phosphate through TDP-D-glucose, this compound is produced. So once we have our compound 6 produced, now it's in the reaction mixture. So it might have some other intermediates, it might have unreacted starting materials, right? So we wanted to purify it. So we used a chromatographic technique known as strong anion exchange. So basically we use the charge of these molecules to separate. So based on the charge, we separated our compound 6 from the rest. If someone can mute, there's someone who is not mute. So if you can please mute yourself, it would be easy for me. Thank you so much. Okay, so this is a chromatogram that I obtained during anion exchange. And because compound 6, if you remember, compound 6, it has this TDP, thymidine diphosphate residue, so it can absorb UV. Because of that, you can see the elution in real time through this chromatogram. So these dotted lines represent the compounds, the fractions that contain my compound of interest. And then I wanted to purify my compound from whatever the salts that is available in these fractions. To get rid of these salts, we used another technique known as gel filtration. So basically, I purified my compound from all the other salts. And then I should also mention that at each of these purification steps, I confirmed the presence of my compound using a technique known as mass spectrometry, or else we call it mass spec. So if you are not familiar with mass spec, it's a technique that is used to identify your molecule of interest based on the mass to charge ratio. So this axis represents the mass to charge ratio and it's the relative abundance. So this mass spec data shows compound 6. So this is the peak corresponding to compound 6. And if you look closely here, we have a ketone group. So in an aqueous solution, it can hydrate and it will result in this compound, which will give a peak with 563.07. So using our proposed pathway with the optimized conditions, I was able to produce and purify compound 6 with a final yield of 80%. And then starting from compound 6 and by adding the remaining 4 enzymes, we tried to produce TDP downosamine in a 1.1 step reaction. That means I added everything together and I incubated the reaction for a certain period to produce this compound. But we faced few challenges. The first thing would be this intermediate number 7 and intermediate 8, they are not stable. Because they are not stable, they tend to spontaneously degrade. So what happens is that at the end, the reaction, instead of it going towards the forward direction, it will go towards the degradation and it will produce TDP and compound 13. So that's not what I wanted. And also, remember I told it's good to have substrate flexible enzymes. So because these enzymes are substrate flexible, apart from it acting on compound 9, it can also act on compound 8. So this is not what we want again. So in this case, it's not good to be substrate flexible, right? So in that case, we saw by mass spec that the reaction drives towards this way, and we see an intense peak that is corresponding to compound 15. So we address these two challenges because the yield of TDP downosamine became low in a 1.1 step reaction. And then we wanted to have a better yield. So we address these two by the first thing is increasing the enzyme concentration of DNMG. So DNMG is an aminotransferase enzyme that can act on this intermediate 8. So remember I told you that these two are not stable, so therefore it can degrade spontaneously. But once we increase the concentration of this enzyme 20-fold higher than the first enzyme, the reaction seems to go towards the forward direction. And also we separated the scheme B into 2 steps. That means first we added the starting material, in this case it's compound 6 with these 2 enzymes, and then we incubated it for a certain period to make sure that compound 6 gets converted to compound 9. And then to the same reaction, we added the remaining 2 enzymes with the co-substrate to produce TDP downosamine. So we call these type of reaction as one pot, 2 step. So you do the reaction in one pot, but you divide it into 2 steps. So then similar to compound 6, I also purify TDP downosamine using anion exchange chromatography. And this is the chromatogram. And this is the mass spec data. So 53009 corresponds to TDP downosamine. And I was able to get a final yield of 14%. Okay, so now we have synthesized TDP downosamine, and then we wanted to synthesize TDP ecosamine. Remember, it is currently produced chemically. So using the same proposed reaction, we wanted to show that this can be used as a platform to produce some other valuable sugar donors by just altering some of the, by just substituting some of the enzymes in the pathway. So instead of using DNMB enzyme, here we used a different substrate flexible enzyme that is known as EPae from a different bacterial species. So EPae can act on, because of its substrate flexibility, it can act on the intermediate 11 and it can produce TDP ecosamine, which is great. In previous literature, researchers have shown that this particular enzyme can be used in vivo to produce TDP acrosamine. So we tried it in vitro. So what I basically did is I repeated the same things. I did genomic DNA extraction, PCR amplified, did cloning, uh, purified the protein, and then used it in the one part two-step reaction to produce TDP eicosami. And interestingly, I was able to reproduce these results in vitro. And here I have shown the mass spec data for TDP eicosami. So if you see here, this peak corresponds to TDP eicosami. So it's basically a stereoisomer of TDP downosamine. Therefore, it has the same mass to charge ratio. So there is a problem. Even though we were able to produce TDP eicosami in vitro, which is great, but there is a problem because in literature it was also reported that this specific enzyme can act on this intermediate as well. So you see the substrate flexibility is not good all the time, right? The substrate flexibility is good in this case because it can produce TDP eicosami, but then because it can act on this, it will also produce another compound known as TDP ristosamine. So TDP ristosamine is again a stereoisomer. So it has the same mass to charge ratio. Therefore, even though from mass spec, I see this peak, I cannot really tell whether it's TDP eicosami, whether it's TDP ristosamine, or whether it's a mixture. So it could be based on the literature, it could be a mixture of these two. Therefore, we have to, in further research, we have to address this issue. We have to find out a way where we can only get pure TDP eicosami. So to conclude, we have proposed a one-part, two-step pathway towards producing TDP downosamine, and using our proposed enzymatic pathway, we synthesized TDP downosamine, and also we synthesized and purified one of the key intermediates, TDP 4-keto-6-deoxy-D-glucose. Even though I did not talk in today's presentation, we also purified compound. Okay, so the numbering should be different here. This should be 6 and this should be 9. Anyways, so we also purified compound 9. So what's the importance of having these two intermediates? So as I mentioned you before, we can use them towards producing other valuable sugar donors, right? So apart from that, we use this compound to kinetically characterize this aminotransferase enzyme, because so far people have not characterized these three enzymes. So I did not present those details today because I thought the content will be too heavy for the audience. So, but I just want to mention that we did use this to do kinetic studies. So basically we found the Kcat, Km values of this particular enzyme using our purified compound. And also we sent some of these compounds to co-crystallize with this, some of these compounds to co-crystallize with this specific enzyme. So it's a collaboration. We sent it to Dr. Rangshi at University of Laval. And also last but not least, we also showed that a proposed pathway can be used to produce TDP eicosami, which is an important sugar donor that can be used to produce epirubicin, right? So what's next? If you remember, the yield for TDP downosamine was not that great. So we have to work on increasing the yield of TDP downosamine. And also because we have TDP downosamine purified, we can use TDP downosamine to do kinetic studies of this particular enzyme. And also we have to address the issue of getting TDP ecosamine with some other byproducts. So currently we are my previous lab is working on it. And the ultimate goal of this project is to engineer a glycosyl transferase enzyme towards producing epirubicin. Because I mentioned to you that even though people have synthesized epirubicin in engineered bacterial systems, the yield is not great. So if we can have POTDP ecosamine and if we can have an enzyme that can act on this sugar donor and the glycan acceptor with a higher catalytic efficiency, would be able to get an increased yield of epirubicin, which will be great. So enzyme engineering, if I briefly tell you, it's a technique that researchers use to improve a specific phenotype. So in this case, we are improving the catalytic activity of a glycosyl transferase to produce epirubicin. Um, Last but not least, I would like to thank my supervisor, Dr. David Kwan, for his tremendous support and his guidance throughout my master's and PhD projects. And also, I would like to thank my amazing lab members and all the funding agencies. And thank you so much for your attention, and I'm happy to take any questions.
So thank you, ma'am. Thank you, Ms. Ifthiha, for the very informative and interesting presentation. Now it's time for the Q&A session. If you have any questions, you can direct it to our speaker. You can unmute yourselves and ask the question, or you can send your question through the chat box as well.
Hello, Ifthiha, can you hear me?
Yes, is this Pasanaya?
Yes, this is Pasa. It was really nice to hear your talk, and it's really exciting. I actually have a question. This is not directly related to your work that you did, but generally. So now you, during your stay here in our department, you were working with plants. You are from plant biotechnology, but now you have expanded your horizon, and you are basically working in interdisciplinary. It's really exciting work. So this is a common question that I see with a lot of younger students. They think that when they select a special or whatever they do, they are stuck in that discipline for the entire the remainder of their career, but it's really not true. So what are the challenges that you had to face when working in such interdisciplinary field, and how did you manage to gain necessary skills to overcome those challenges? So those are my questions. Thank you.
Yeah, that's a great question. Yes, like if you look at my scientific career, like I started as a plant biotechnology student, and today most of my research it's more towards chemistry, even though it has biology. So yeah, so my PhD lab, my supervisor, he's a biochem prof. So he had this idea where he wanted to merge biology and biochemistry together. So that's how I ended up with this project. And initially, yes, it was a challenge because I mean, many of the techniques that I performed by myself, we did not learn it during our undergrad because I wasn't specialized in chemistry. But then, because I had the basic chemistry knowledge, because during your first and second year, we all have to learn like analytical, inorganic, and also the general chemistry part, right? So it helped me a lot to understand certain concepts and techniques. And then, yeah, that's how I basically uh, like, even though it's a challenge, that how I basically address those based on my previous knowledge. And also I should mention that my supervisor, he's so supportive. So he will walk me through each and every step if I'm not clear or anything. So yeah, but I mean, just this is just to everyone out there. I mean, don't don't think that if you choose a special like a specific area during your undergrad, you have to be in that field throughout your entire life. Let's say if you are a plant biotechnology student and if your project is on some plant species, you don't have to end up your life in that particular plant. I mean, if you are interested, yes, obviously you can do that, but then there is a big world out there where you can explore. And I mean, the professors here, they are very supportive, and same as the professors in University of Colombo, everyone is very supportive. So if you have a basic knowledge, I mean, you can quickly catch up everything. So I would like give a piece of advice, like whatever you are doing now, do it correct. If you are in your first year or second year, like do all the subjects like to the fullest potential that you can, so that your future will be so easy.
Thank you, Ifthiha. That was a nice explanation, and I wish you all the best for your future work as well.
Thank you so much.
So will there be any more questions from the audience?
Hi, I'm Tanusha here.
Yes, Tanusha.
Hi, Ifthiha. So it was a nice presentation actually. So like, if I added more just to Pasanaya said, like, even I'm from a plant science background. So I did like, for my fourth year, it was like totally plant identification and systematics and all. Then again, I ended up in a research similar like to like yours, right? I have just a small question to you. Now, in this streptomyces, this anthracycline synthesis pathways. So, have you like, did you find any specific conditions which these streptomyces more synthesize more anthracyclines, like any in you mean in any culture conditions that you did, like any nutrient deprivations or any, you know, like streptomyces go through many states, like vegetative to non-vegetative states, right? So during that transition, so is there any specific stage that this streptomyces synthesize this anthracycline in their cells?
Yes, Tanusha. Yeah, that's a good question. But to be honest, even though all of these genes are from streptomyces, I never cultured them.
Okay.
So yeah,
Yes.
so basically because all my research was in vitro, so I basically took the
Yes.
gene and then I purified it. But
Yes.
I remember in literature there were few conditions, few culture conditions actually,
Okay.
that was reported to produce these anthracyclines in very like in good yields, but it's not very good per se, that it cannot we cannot use the produce anthracyclines from that organism
Yes.
to the current need. It cannot fulfill the current need. That's why even for doxorubicin. Doxorubicin production is good so far in bacteria, in streptomyces, but doxorubicin also, even though it is produced in streptomyces, it is also currently produced semi-synthetically because it cannot meet the requirement of today's world. But there are obviously conditions. So what people were more interested is that they try to like work on these genes. They try to, you know, knock out certain genes. And yeah, and another big challenge in these organisms is that there is a certain limit, there's a threshold that these organisms can hold, because these are like doxorubicin and daunorubicin, we call them as anti-cancer agents. So it can harm the cell once it reaches a particular limit. So in that case, it's not so ideal to produce it in these organisms. But researchers, people are working on it.
Okay, right. Thank you for the answer. And like, it was a nice presentation.
Thank you, Tanu.
Hello, Ifthiha. Can I ask another question? Hello.
Yes, for sure.
So can you comment? So you have used some aspects of synthetic biology in your research as well, right? So can you comment on the current stage of research in synthetic biology and how it's used in general for scientific research? Because I remember like there was Craig Venter who was like a prominent pioneer in that area. So how does that improved in the last couple of decades and how it involves in your research?
Yeah, that's a good question. So my PhD, I did my PhD at Concordia, and like we had some amazing professors who worked in the area of synthetic biology, including my prof. So basically, like in today's world, like mostly to the best of my knowledge, because what I saw from those professors is that many of them are trying to like engineer enzymes and, you know, use it towards either producing not novel, I mean, as an alternative to the chemical production. So they basically, some of the projects that I have encountered is like some of the projects are on production of therapeutics, like in the case of epirubicin production, where the PhD student that my professor recruited recently, she's working on engineering an enzyme to produce epirubicin in a biological system. So similarly, like there are a number of projects that is going on to produce like as a replacement to the chemical methods. And also I know that some people are working on plastics. So they're trying to synthesize plastics biologically, which is cool. And also some are working on biofuel projects as an alternative to this gasoline fuels. So yeah, like that's what I saw, but I know that there are a lot of research that is going on using these synthetic techniques where they apply these gene editing tools and everything, where they apply CRISPR-Cas9 systems and everything to get like better outputs.
How is thank you for the answer. So how is the connection with the commercial industry? Like are your research in your university, are they connected with, do you have any commercial aspects in it? Do you produce any products where you send it to a market? Or how is that pipeline in Canadian universities, at least in general?
I mean, in my university, like all of these research were at its beginner level. So many of the profs, they were recruited like maybe 5-6 years ago. So still it's not at the point of commercialization, but I believe that there might be other projects that have reached that level in Canada, but I mean, I'm not aware about it though, but I could always look into that.
Thank you very much, Ifthiha. Thank you.
You're welcome.
So it seems that there are no more questions. I would like to thank the speaker, Dr. Ifthiha Mohideen, for her valuable time and the knowledge, experiences that she had been sharing with us. So I think our audience got the maximum benefit from your presentation. Once again, thank you very much, Ms. Ifthiha Mohideen, for being the resource person of today's Sci Talk webinar. Finally, on behalf of the WhatsApp, I would like to thank all the lecturers of the Department of Plant Sciences, Faculty of Science, University of Colombo, and especially to Dr. Anushka Vikramasuriya, Senior Treasurer of WhatsApp. So, this is the end of 9th Sci Talk webinar session. Good day to all, and thank you for joining today with us. Let's meet again at the next webinar of the Sci Talks. Thank you.